Hey everyone, I have a special treat for you. This paper is really fantastic and sort of goes along the lines of the last psilocybin production in yeast paper. This is out of a group from UC Berkeley, and it's in the journal Nature, which is a very big journal, but it's just a letter, so it's a little bit of a smaller paper. And I have an intimate relationship with this article personally. Over the past 4.5 years or so, I've probably read it about a dozen times because in the beginning of my PhD, I'm just about to finish my PhD this month, but at the beginning of my PhD, for about 1.5 years, I was trying to make cannabinoids in Eurolitica. And so reviewing this paper is going to be really fun because I'll be able to provide some insight of like how actually hard this is to do, which the answer is very. And also the feasibility of this at an industrial scale, or potentially if you were to take home a strain of this yeast, how useful that would be to you. Great, let's dive in. I'm super excited. The paper is called Complete Biosynthesis of Cannabinoids and Their Unnatural Analogues in Yeast. So this unnatural analogues is sort of the last third of the paper, and I'll only talk about that briefly, but it is interesting. So complete biosynthesis, also known as de novo biosynthesis, meaning from just very basic ingredients you can feed the yeast, so sugar usually. So this was all done in, here, let's start here. The psilocybin paper that I reviewed a couple of days ago, it was a very, very, very traditional metabolic engineering paper. This is pretty traditional metabolic engineering, but with a few twists. So we'll talk about that. It's also by an author named Jay Keasling, and he is potentially the most well-known metabolic engineer in the world. At the very least in the top 3, right? So this is really important work by a very important lab. Like a lot of metabolic engineering papers, they set up their argument here for we can make these compounds other ways, but why don't we make these compounds in yeast instead? So they say low in planta abundance of the nearly dozen of all the dozens of known cannabinoids. So there's actually over 100 cannabinoids known naturally in the plant. And they say low in planta abundance. And you might be thinking a lot of flour now has 30% cannabinoids, which is seemingly really high. Relative to the whole plant, that is kind of small. But mainly I think what they're talking about here is that the minor cannabinoids are in very low abundance in the plant. So yes, we get a lot of THC. Yes, we get a lot of CBD in separate strains, of course. But THCa, if you've ever heard of that one, are we really getting a strain that makes a lot of that? No. We have a lot of strains that make a little bit of it, but not a lot of it. So that's their argument here. Again, this is in baker's yeast or brewer's yeast, Saccharomyces cerevisiae. It's been domesticated for thousands of years by humans and really well studied in academia. They do it from the simple sugar galactose. So again, this is de novo. Galactose is pretty similar to glucose. It's a monosaccharide, so it's just one sugar unit. De novo production, and they also showed that due to the promiscuity of the enzymes in this pathway, they're able to feed different fatty acid analogues to the strains to produce different cannabinoid analogues. So we'll talk about that more later. There's a lot to unpack there, but I'll get to it. And through that, they're able to further expand the accessible chemical space for these different cannabinoids. So basically, they can invent new cannabinoids through biology. Let's move on to the first figure, and then we'll break everything down. So metabolic engineering has to do with engineering these metabolic pathways of different biochemicals, right? So here's the pathway for cannabinoids, either in plants or in yeast in this case. You have some basic carbon source, so some sugar typically, that's converted to acetyl-CoA, which is a very predominant cell precursor to other molecules. And this can be converted to 3 different things that are relevant here. One is moving it through the mevalonate pathway to produce geranyl pyrophosphate. 2, converting it to malonyl-CoA. And 3, converting it to hexanyl-CoA. So then this malonyl-CoA and hexanyl-CoA can come together with two of these CS enzymes, so cannabis sativa. When enzymes are named, usually the first letter is the genus and the second letter is the species. If you notice that the first is capitalized and the second is lowercase. So these are cannabis sativa enzymes. So there's two cannabis sativa enzymes that take malonyl-CoA and hexanyl-CoA and make olivotolic acid, this compound here. Then GPP and olivotolic acid are converted by this CSPT4 enzyme, so another cannabis gene, into cannabigerolic acid, or CBGA. And then CBGA is, it's considered like the mother of all cannabinoids, right? And the reasoning is it's sort of like the Pokemon Eevee. You give it a different enzyme and it will make a different product. So if you give it CBDA synthase, then you get CBDA. And if you give it THCa synthase, you get THCa, which are both precursors to THC and CBD, right? So then you add those enzymes to make the cannabinoid that you want. CBGA is also a cannabinoid and also has its own uses, but it's the mother, and so you convert it to other products. And then you have this last step that says delta T, which is a change in temperature, referring to an increase in temperature to decarboxylate these carboxylic acid cannabinoids. So this COOH group is a carboxylic acid group, and that needs to be removed to make these compounds in their most active form. So this is why you have to smoke cannabis. You can't just take a flower and eat it and expect to have effects, at least not the same effects. And then same with CBDA. Removing this carboxylic acid group makes CBD. And same thing with CBG. If you want CBG and not CBGA, you have to heat up this molecule and remove that carboxylic acid group. So that's, again, why you have to smoke cannabis typically, or if you want to make like an edible product, you have to heat up the flower beforehand or in oil or something like that. You have to decarb it, you have to decarboxylate it, right? Okay, so that's the pathway. When I was trying to make these compounds, I didn't add all of these enzymes that they're listing here. I just added CSTKS, CSOAC, CSPT4, and I only tried to make CBDA, so I added CBDA synthase. And even if I added hexanoic acid, or I added hexanyl-CoA, or I added olivotolic acid, or added CBGA, I never was able to get any enzymatic conversions to happen. And as we go through this paper, you'll see that these enzymes are just very, very inefficient. And this inefficiency may predominantly be due to moving these enzymes from plants into yeast. So in the psilocybin paper, we're moving from a fungi to a fungi, from Psilocybe cubensis to Saccharomyces cerevisiae, the hyphal fruiting fungus, to yeast, right? In this case, we're moving from a plant, a separate kingdom of life, into a yeast, another kingdom of life, right? And so potentially these enzymes don't translate as well. This is not always the case. This is just my speculation that's the case. Or, these enzymes are just making secondary metabolites, so maybe even in the cannabis plant they're really low activity. When I was trying to make cannabinoids in the other yeast, Eurolitica lipolytica, I was doing it partially because Eurolitica lipolytica is known to make a lot of this precursor acetyl-CoA. And that's sort of one of the big movements in biotechnology right now, is we're able to work in these non-conventional yeasts that specialize in a particular thing. So Eurolitica might grow a little bit slower than Saccharomyces, and it's a little bit stinky too, but it produces a lot of this acetyl-CoA, so it could potentially produce a lot of cannabinoids. In a traditional metabolic engineering article, they'll say, okay, we want to make this chemical, and so we made this strain that produces the first chemical in this pathway. And then we want to make the next chemical in this pathway, so we made a new strain that makes the next chemical in the pathway. And so that's what they're going to do here. And then once they make their chemical of interest, they'll say, okay, how do we improve this? What is the rate limiting step in this pathway? Is it this CSPT4 enzyme or this CSTKS enzyme? We'll find out. Here's what the rate limiting step is, and here's where we're going to improve it. And so they do this in this paper by naming their strains YCAN 01 through whatever number strain they're working on, right? So for the first strain, they're trying to just produce olivotolic acid. And so they added the CSTKS and the CSOAC enzymes. They actually created a linker between these two enzymes and put them in together. And their reasoning there is that the steps are sequential. So you need the CSTKS and then you need the CSOAC enzyme. And so they said, let's just physically put them closer to each other when they're manufactured in the cell, and that might improve the synthesis of olivotolic acid. There's a more recent article that showed that this is not the case and this is not the best thing to do, but it was a good thought. They make this strain with CSTKS and CSOAC, and they find that it produces 0.2 milligrams per liter of olivotolic acid from galactose. Now, this has been done before, so it's not that impressive, but they're making it from the sugar, and they're making this new molecule that isn't natively made in yeast. And then they move on and they say, okay, well, if we feed an amount of hexanoic acid, the precursor to olivotolic acid, we get a 6-fold increase in olivotolic acid production. So now we're at about 1 milligram per liter of olivotolic acid. And so you might be able to say here that one of the rate limiting steps is the cell producing hexanoic acid. That's just an example. They didn't do any engineering to increase hexanoic acid, but that's just an example there. The next step here is to produce CBGA. So they need a prenyltransferase enzyme, or more broadly, they need a geranyl pyrophosphate olivotolate geranyl transferase, or GOT. That's a mouthful. Scientists come up with great names. Basically, the name of that means they want to put this GPP and olivotolic acid together to make CBGA. And this is one of the big novelties of the paper. So there's previous papers that have done basically all of these steps except for this one. You know, it's important that they figure out how to do that next step. So in order to do that, they tested, I believe it's 8 different GOT type enzymes. A lot of them were from the cannabis sativa plant. And so this is what this is saying here. So they were searching for candidate prenyltransferase enzymes. So prenyltransferase has the activity that can solve the GOT function. And what they ended up finding is that there's only one that worked for them. So this strain that expresses CSPT4 in the YCAN14 strain produced detectable amounts of CBGA. So they got 136 milligrams per liter. But in this case, it was fed libitolic acid, like 1 millimolar. Yeah, 1 millimolar libitolic acid. So they were giving it the direct precursor, but they were getting a lot. So another non-rate limiting step here is this CSPT4 step. So if you give the cell plenty of libitolic acid, you're going to get a lot of CBGA. And so this CSPT4 step is not rate limiting then. Great to know. Another mystery solved. That's fantastic. They did a little bit more characterization of it. And so they also found that when they put the CSPT4 enzyme in the strain that they had previously from galactose, they were able to make a little over 1 milligram per liter of CBGA. Now, let's talk about titers for a second here. So 1 milligram per liter, that probably sounds like a small amount to you. That's because it is a little bit. So, you know, if you're in a dispensary or something like that, and you're looking at THC in edibles or vapes or whatever, a dose typically is probably somewhere between like 5 and 50 milligrams, right? And a liter is like this big, right? So imagine making at most 20% of a dose in a volume this big. That's not going to be profitable, right? Really, in an industrial biotechnology setting, if you're making a liter, whatever product you're making in a single liter, you should be looking at $1,000 of product per liter, on that order at least, because of the upstream processing. So like doing the genetic engineering of the strain, that costs a lot of money. The employees cost a lot of money. They're very, specialized employees. And then the downstream processing, taking that liquid and then purifying the compound that you want, also is very labor intensive, costs a lot of money. Optimization may be difficult. And so really in 1 liter, you want like $1,000 or thousands of dollars of your product in order to make it profitable. Okay, so 1 milligram per liter of your cannabinoid is not going to cut it, most likely. They're also saying here, they're flexing on us a little bit. So they made CBGA, great. Now they're flexing on us here and saying, look, nobody has ever made THCA or CBDA in yeast, so in vivo, in the living system, from sugars, right? So they're going to add either CBDA synthase or THCA synthase to make these cannabinoids and really flex on us, right? One thing that they had to do with these cannabis sativa enzymes is they had to replace some of like the signaling parts of the protein with other signaling molecules. So like for the CSPT4, for example, they had to we removed predicted N-terminal plastid targeting sequences. So on the CSPT4, there was an N-terminal. So N-terminal just refers to which end of the protein. So on one of the ends of the protein, there was this plastid targeting sequence, which doesn't make sense in the context of yeast because yeast don't have plastids. Plastids are plant-specific organelles. Okay, so we had to remove that tag, we being not me. And then for these THCA synthase and CBDA synthase molecules, they had to replace the N-terminal secretory peptides, which would tell the cell to move the protein outside of the cell, which isn't very useful if you want to make your protein within the cell, which is what they did. So they had to remove that and add a vacuolar localization tag, which allows the cell which allows the protein to stay within the cell. Okay, and then they made this new strain, and when they fed hexanoic acid, they made about 1 milligram per liter of THCA and 4 micrograms per liter of CBDA. This is tiny amounts. And weirdly, they found that when they just fed the cells galactose, they actually got twice as much THCA and the same amount of CBDA. That's saying that in this case, the hexanoic acid is not a limiting factor. They made THCA and CBDA. That's fantastic. Another big novelty of this paper. They state some promiscuity of at least some of the pathway enzymes. So this is what I said at the beginning of this video. Promiscuity in the world of enzymes, at least, refers to an enzyme being able to accept different substrates and produce different products, right? So they said, what if instead of feeding the cells hexanoic acid, we fed them a shorter carboxylic acid like butyric acid? So what that does, well, sort of the motivation here is they found that they also produce 2 new cannabinoids, new in yeast at least, THCVA and CBDVA. So what are those? We can look at them here. Actually, they don't have a diagram down here, but you can imagine here, instead of feeding hexanoic acid, if we feed butyric acid, which has a 3-carbon side chain instead of a 5-carbon side chain, we'll end up with a 3-carbon side chain here, and we'll get butynyl-CoA instead of hexanyl-CoA. So you imagine this is 1, 2, 3, 4, 5 carbons on the side chain. This is also a 5-carbon side chain and a 5-carbon side chain and a 5-carbon side chain. If instead we have a 3-carbon side chain that we start out with, then we'll get varinic acid instead of a levitolic acid. We'll get CBGVA with just 3 carbons instead of 5. We'll get CBDVA with just 3 carbons instead of 5. So, they found that these enzymes in this pathway are promiscuous to producing other chemicals, which is interesting and potentially very useful. So they found that they made all of these sort of alternate reality cannabinoids with the V in the name, with the 3-carbon side chain. They step back for a second and they want to increase the titers a little bit. They basically find that this CSTKS, CSOAC step is rate limiting, so this step here is rate limiting, and so they overexpress those enzymes hoping to make a little bit more, and they get a 3-fold increase in titers. Great. Okay, back to the promiscuity thing and the titers that they were achieving here. So this is chromatography. Specifically, this is liquid chromatography with mass spectrometry, so LCMS. And like I explained in the psilocybin video, basically you have some way to separate your sample, and then on the other side of the separation you have a detector. So you send your sample in, for example, THCA makes it through the separator, and you have a detector that sees that molecule as a peak. The area under that peak is directly proportional to how much compound is in your sample. And if that peak is present, that probably means you have that compound. And every single time you run that method, it will come out at the same time. So if you run your standard and you see your standard at 11.4 minutes, and then you run the culture media, so you run the cells that have grown from the YCAN42 strain, and you see a peak at the same time, then you probably have production of THCA in this case. So they're basically demonstrating here, like, yes, believe us, but also here's the data. Here's THCA being produced, here's THCVA being produced, and then the CBD equivalents as well. All right, so they took advantage of that. Well, first of all, the best yield that they got of THCA was 8 milligrams per liter, and then 4.8 milligrams per liter of THCVA. And the significance here is that, in the cannabis industry, maybe 20 years ago, CBD was very expensive, believe it or not, maybe $10,000 a kilogram in a wholesale setting. Now it's closer to like $500 per kilogram because everybody and their grandmother makes it now, right? Same thing with THC. Less time ago, THC was maybe $30,000 a kilogram, and now it's closer to probably $1,000 per kilogram wholesale. So what happens is there's this big push from the market for this new cannabinoid, and then the providers of that compound have to catch up to that supply, and then they overproduce and demand doesn't change, or maybe even demand drops off, and you have all of this supply, not enough demand, and so your price plummets, right? This also happened with like CBN and THCV most recently. So THCV, just like a year ago, was like $30,000 a kilogram because no one had any of it. And now you go to any dispensary or any smoke shop if you're in a state where cannabis is illegal, and you see THCV products, partially because the federal government hasn't said that it's illegal, right? But it still is psychoactive. So anyway, this is what happens. There's huge demand. Since it's a plant natural product, people have to breed new plants or develop new chemical processes in order to make the compound. Whereas a big advantage here with yeast is potentially, even though these titers are really low, this is maybe, $5 or $1 worth of THC in a 1 liter vessel. That's not very good. But a year ago, this 5 milligrams in 1 liter of THCVA might have been worth closer to $100 or something like that. And so if you're working with biologics, this is one of the big advantages, is you can quickly pivot to making a different product, especially when you're working in a class of compounds like cannabinoids. You just flip one enzyme, say, the market goes from CBD to THC, you just flip from CBDA synthase to THCA synthase. Easy. Or it flips from the V-style compound, and so you start feeding the cells butyric acid instead of hexanoic acid. And so this is one of the advantages here with this biotechnology approach to producing cannabinoids. And the Keasling group was able to demonstrate this even further by feeding this cannabinoid producing strain 19 different fatty acids of various chain lengths, branching and saturation degrees, and they were able to make most of them in this strain. So instead of butyric acid or hexanoic acid, they can feed all of these different molecules and make new cannabinoids, right? So maybe some of these have been studied, maybe they haven't been studied. Maybe they have, like a sort of FDA approval style pharmaceutical effect, or maybe they have sort of the more holistic approach that a lot of cannabis users go by. But either way, they might have a new effect that has never been seen before in nature or has never been produced in high enough concentration to be studied properly. So this is really cool. And on top of this, they were able to feed a certain carboxylic acid that had a particular side chain that allowed them to do further chemistry. So now you're producing these precursors to make sort of semi-synthetic cannabinoids as well, which is really cool because we might want to study those and they might have some profound effect. even if you're of the party that like, oh, we need everything to be natural, whatever, you can have that. You can have your natural product, but some of these molecules may be derived from cannabinoids. You know, there might be some future molecule that's derived from cannabinoids or derived from the cannabinoid pathway, but it's like an anti-cancer drug or something like that, or it treats Alzheimer's or something like that, right? And so in the pharmaceutical pipeline, this is important as well. So anyway, I think that's all I have to say on this paper. It's a really cool one. You should go read it. If you want to get into metabolic engineering, this is a really good way to. Hopefully, I've sort of tricked you into my sort of van of metabolic engineering with drugs, and you're able to learn a little bit about metabolic engineering through this article. If you did, please make sure you subscribe to the channel, like the video. Let me know in the comments below what you want me to cover next. I'll see you in the next one. Bye.